Oh, hi. Welcome to KC Science from Bigelow Laboratory for Ocean Science. Today we have Emma Cold, working under Dr. José Fernández Robledo, to explain her project. Let's go find her.
Oh, hi. I'm Emma Cold, and welcome to my lab. Did you know that 453,000 people are affected by malaria each year? And treatments often spoil before they even make it to these third world countries. And there's actually really no effective vaccine for it because it's such a complex parasite. But I think we have the solution right here along the coast of Maine. Let me introduce you to Perkensis marinus.
Perkensis marinus is a parasite that affects oysters and causes death 1 to 2 years after infection.
Now in 2014, a study was shown feeding live Perkensis marinus to humanized mice.
Humanized mice are genetically modified to have the immune response of a human.
Well, that being said, after the feeding was done, a crazy thing happened. Nothing. It didn't cause a stomach ache to the mice, it didn't cause a headache, but the only thing it did cause was an immune response, meaning it made antibodies to Perkensis marinus. It made an army against it, but the mouse won. And that's exactly what we look for in a vaccine. So I'm in the early stages of taking this parasite and genetically modifying it into a vaccine. You're probably asking, exactly how do we do that here at Bigelow? Well, the answer is literally quite shocking.
Cell electroporation is using electricity to open up a cell membrane to deliver chemicals or DNA.
I'm introducing a plasmid to Perkensis through electroporation. A plasmid is like a vehicle that delivers very important information to Perkensis, so Perkensis can express that information. And this plasmid contains genes that are shown to be very good candidates for malaria vaccines. This plasmid also contains a green fluorescent protein, or GFP. This acts as a marker because it glows when you look at it under UV light. Since GFP is in the plasmid, if we see GFP, we know that the vaccine genes are also being expressed. If this works in the long run, we feed the genetically engineered Perkensis with the plasmid to the humanized mice. We check to see if the mice had an immune response specific to the malaria vaccine genes.